الرحيم. السلام عليكم. بنبتدي السيشن الثاني في جلسات المحاضرات لليوم الأول من مؤتمرنا الثاني عشر للعلوم الصيدلية. برحب بزميلي دكتور هشام توفيق. هذا الجلسة المحاضرات بتشمل على 4 محاضرات هنقولهم، وبعدين بعد نهاية المحاضرات إن شاء الله هنبتدي نعمل الأوبن ديسكشن. أول محاضرة بيقدمها لنا بروفيسور إيمانويل كورنيلو من فرنسا، بروفيسور of Cell Biology Montpellier Cancer Research Institute University of Montpellier France. عنوان المحاضرة Deciphering Interactions Among Tumor Microenvironment from Transcriptomic Data. Welcome, Professor Emmanuel. You are the first in this session. Welcome. The second lecture will be led by Professor Mustafa Kamel El-Awadi, Professor of Molecular Genetics, National Research Center, Cairo, Egypt. The lecture entitled Genome Editing Technology in Drug Discovery and Treatment of Rare Liver Diseases. دكتور هشام.
أحب أرحب بكم مبدئيا في المؤتمر العلوم الصيدلية، وفي السكند سيشن اللي هتبقى فيها التشيرمان الدكتورة تهاني، وأنا إن شاء الله هكون معاها بإذن الله، وهتبقى الثيرد لكتشر فيها أو التوبيك فيها هيبقى للدكتور يوسف عبد الله بن جوردن، he is associate professor from Department of Pharmaceutics, College of Pharmacy, King Saud University, and he will talking about the effect of hyperlipidemia on the deposition of dronedarone in rats. And further, it is the last lecture which will be for Dr. Tamer Mustafa Saqr. He is associate professor from radiopharmaceutical chemistry, Egyptian nuclear research reactor, and he will talk about the nano-radiopharmacy approach for tumor cyanosis. We will begin with Professor Emmanuel.
Okay. First lecture, please.
First to thank the organization committee to let me present today the work we performed over the last year on the deciphering of interactions in the tumor microenvironment using transcriptomic data. This work has been performed at the Cancer Research Institute in Montpellier called IRCM. This work I'm working under the supervision of Professor Jacques Collange, which headed the bioinformatics and system biology of cancer team. The idea today is to present you through different work we perform, how we succeed to, and not only new, but how all the recent research that have been developed on transcriptomic data, that is on the study of the whole mRNA expression, how we succeed to move from the description of tumor diversity generally based on what we call the hallmark of cancer, so this accumulation of modifications inside the cancer cell and the different all the diversity of evolution which are possible inside the cell, to the description of the tumor microenvironment, that is the tumor cells and the other cells that are also present in the tumor. And the idea is that tumor microenvironment is in fact the result of hijacking by the tumor cell of the different cells. So common cell types that exist in the body, but which in fact will lose their functions, will acquire new phenotypes to help the cancer cell to continue its development. So among these cells, we found fibroblasts, so-called cancer-associated fibroblasts, or macrophages, so-called tumor-associated macrophages. So you see through these names that these cells and cell types have been modified. We also will find many lymphoid cells, so immune cells that will infiltrate the tumor, which at the beginning are there to fight against the tumor, but we'll see they are progressively exhausted. So we need to reactivate them. And in fact, this is the great success of the recent therapies based on immune checkpoint inhibitors, these monoclonal antibodies, which have been developed against certain ligands and receptors. And we have also, which is important, the extracellular matrix, which play an essential role in all this communication, in combination with the secretions of many cytokines, so chemokines and growth factors. So you have to imagine for each tumor, and this is the general hypothesis now, is that for each tumor we have a continuum of tumor immunity, from inflamed tumor, which are highly infiltrated with lymphoid cells, and which are also presenting high expression of the interferon gamma pathway. And we have something also where we have quite a lot of immune cells, but the cells are at the periphery of the tumors, called immune exclude cells, which is characterized by high expression of TGF-beta. And then we have these immune desert tumors where there are strictly very few immune cells present in the tumor, and which is characterized in many tumors by high expression of the Wnt beta-catenin pathway. So the composition of the tumor generally addressed using different techniques. So the first one is fixation of the tumor and staining for immunohistochemistry, or also for immunofluorescence analysis. Bulk RNA-seq is used and has been developed for many years, first using signatures from different softwares like CyberSort, or so what we call deconvolution software, based on cell signatures from single cell culture, sometimes that were isolated by cytometry, but very recently single cell RNA-seq has been very successful for the identification of new cell signatures that are used to identify and quantify cells from bulk RNA-seq. So bulk RNA-seq is the whole RNA you extract from a tumor. Single cell RNA-seq is the RNA sequencing you perform on each individual cell in a tumor. And this is very interesting because this new type of data set has really improved a lot the description of tumor microenvironment and even tumors and radio development and so on. You can identify cells, but you can also identify cell state. So based on the activation of specific pathways inside the cell, you can identify cells which are the same type, but in different stages, either of development or in the response to specific stress, for example, or whatever. And we recently developed an algorithm called single cell signal error, which is there to describe and quantify ligand receptor interactions. And this work has been recently published by our team and is associated to the development of a database, that will be used in the presence in the studies that were presented, that was used in the study I will present today. So the first study I wanted to present to you today is the study of salivary gland carcinoma. This is rare tumors from the salivary gland, and so they represent only 2% of these tumors, and they are characterized by high aggressiveness and mortality rate. And why we're interested in these rare tumors is because these tumors are so rare that it's difficult to develop new therapies for patients. So the idea is to infer from what is known on other tumors, the new therapeutic options for our patients. So we start from the description at the level of genes by using differential gene expression. Here you have the top an unsupervised clustering based on immune genes, and which are compared with so normal tissue. Here we have 2 normal tissue in the study, and the rest are SDC, so salivary duct carcinoma. And there are 2 origins of the RNA-seq data. We analyzed 10 patients in Montpellier, and we add to this patients from the Sloan Kettering, which were RNA-seq data that was publicly available. And when we then look after deconvolution using MCP control algorithm, when we look to the composition of the cell inside the tumor, we realize that indeed we found immune-rich tumors on one side and immune-poor tumors on the other side. And this distribution fits quite well with the level of expression of immune genes. And using immunohistochemistry, using with CD8, so T-cell specific antibodies, we could in fact found the 3 phenotypes described previously. So the immune-infiltrated tumors, the immune-excluded tumors, and immune-poor tumors. Now, when we further analyze the level of expression of interferon gamma, we realized that this expression was in fact associated with low prognosis for the patients. And you have to understand that interferon gamma is responsible for macrophages proliferation inside the tumor. And here, when we look to the different phenotype of macrophages, we realize that many macrophages were in fact M2 macrophages, which are known and were confirmed here to be of bad prognosis for patients. So it is here a very classical crossroad that is also present in SDC, where you have T helper cells that produce interferon gamma, and which are responsible for the proliferation of macrophages. But in the case of SDC, some macrophages in some tumors, or in fact in many tumors, will be polarized to become M2 macrophages, which are responsible for the production of TGF-beta. And this explains mainly the main problem we have to treat patients having this type of tumors. And meanwhile, what we also characterize is the presence of this dystoplasmic stromal reaction, which is in fact very strong in SDC. And we also could characterize that somehow this desmoplastic response is associated with bad prognosis, which is not necessarily the case in all tumors. But here we clearly have something where we have this crossroad where we produce macrophages, and in some macrophages, in some tumors, the productions of this extracellular matrix will lead to specific proliferation of fibroblasts. And in some cases, this proliferation, in fact, will promote tumor progression. So applying the ligand receptor detections using this ALERDB database, we could characterize the presence of the main ligand receptor pairs known to interact with, known to be responsible for immune checkpoint and to inhibit T cells. And So TIM3, galectin-3 was highly expressed in immune-infiltrated cells such as PD-1, PD-L1, and CTLA-4, as we combined with CD80 and CD86. In one, we could analyze one patient for the co-localization or side localization, I would say, of the different markers of the ligand and the receptors in the tumors. And what we could observe is that about 80% of the PD-1 cells were also associated with PD-L1, and at the level of about 80%, which is also the case for CTLA-4 with CD80 or CD86 cells. But in the case of TIM3, they were nearly 100%. All the cells were in fact presenting co-localization between the two markers. But when we look to the cells that express these ligands, so PD-L1 that inhibit PD-1, which is so PD-1 at the surface of the T cells, and PD-L1 is in 30% of the case associated with macrophages. But in the early cases, this is the cancer cell which expressed PD-L1. And for CTLA-4, we found CD86 mainly associated with macrophages also, so inhibiting T cells. The situation for TIM3 and galectin-9 was slightly different, where we could characterize a pathway that was not described so far, where macrophages and NK cells both express TIM3 and galectin-9 at their surface. So, and maybe also initiating cross activation and also maybe autocrine regulation. The second study is also based, we also would like to study and characterize the tumor microenvironment in a rare tumor. It is B-cell lymphoma localized in brain. So this is a very specific tumor, which is quite rare. It's 4% of the brain tumors, and it's very difficult to target because of the presence of the blood-brain barrier. There are currently many immune therapies going on, especially PD-L1, anti-PD-L1 therapies now starting on in this tumor. So it was very important to provide a new up-to-date description of the tumor microenvironment and possible effects of this therapy, especially because of this. So better description of this tumor, especially because so far they were considered as a single type, and in fact, when we run the same analysis as we do before for SDC, we could in fact identify once again poor tumors, intermediate tumors, and immune-rich tumors. But what we do here in this study is we add, and so it's 50 students who had the idea to select HLA genes and see how HLA gene will group the tumors. And in fact, we can find 4 different types. So a group of tumor, which mainly immune-rich tumor that expressed both MHC1 and MHC2 at their surface. And then we have 2 groups, one expressing only MHC1, so MHC1 genes, and so MHC2 associated with so here in white is when we're below the median, so it means down expression. So we had MHC2 only, or MHC1 only, or none of them. And we see that the tumors expressing none of MHC1 and MHC2. So, so tumors that are not presenting epitopes at their surface are indeed so tumor that do not interact and recruit the immune cell, which is quite consistent in fact. But what we further could characterize is, despite the fact that according to this, so HL1 expressing tumors are indeed of good or better prognosis, and that's so we could observe earlier relapse in tumors expressing none of the HLA system. We could also found that MHC2 trigger CDS plus and Th1 expression, and that this is directly related also with the expression of interferon gamma pathway and extracellular matrix. But what we see here is that we're in a slightly different situation as in SDC. But we have the classical pathway where so Th1 produce interferon gamma, which induce macrophages proliferation, which modify extracellular matrix. And in fact, we're in an environment where macrophages and maybe tumor cells will inhibit the T cells, but in some tumors. And so this is this crossroad that's quite straightforward, but we have no polarization to M2 in this case. So it's much more favorable for the development of, for example, anti-PD-1 or anti-PD-L1 therapies. When we look to the oncogenic signaling pathways, we found something which is consistent with this hypothesis. This is the presence of STAT3 and interferon gamma pathways that were highly correlated with immune-rich tumors. Here, what you have in this first figure, in the first part of the figure, this is scores for pathways. So it's different, many genes, and we score the expression of the genes in the pathway. Oops, sorry. And we see that it is, so the expression of these pathways are tightly related with rich tumors. For TGF-beta, which is something intermediate, with which suggests that maybe we have something similar with an exclusion mechanism, whether it was never described, and we could not describe that or see that in this way as it was for SDC in these PC and SL tumors. And we found again the Wnt pathway tightly related with immune poor tumors. And what we could observe using immunohistochemistry is that so presence of tumor-associated macrophage with PD-L1 is in fact of good prognosis for patients. You have to consider that most of these patients received also treatment based on rituximab antibody. And this clearly show that we're in a very different situation as in SDC. So altogether, I hope that through these two examples, have described you the great interest of using RNA-seq to identify so the composition, to establish the composition of the tumor microenvironment at the cell level, to see also the main oncogenic pathways that are expressed. And now I would like to insist on the fact that also we have very interesting drugs to put in front of these descriptions. It means that now we can start developing integrated therapeutic approach where we integrate tumor description with new therapeutic options for patients. And I would like to finish my presentation and to conclude on this full pipeline where you see that we have different algorithms now available in our team to describe and confirm the role of the tumor microenvironment, to identify the oncogenic pathways which are correlated with this, the presence of this microenvironment. We can classify samples between rich, intermediate, and poor immune infiltrate. And we also could identify some resistance mechanism, which are very important to decide whether a patient could benefit from a certain type of therapies, which is, for example, the MHC1 and 2 expression. We have this ligand receptor database and algorithm to study cell-cell communications and to really emphasize the patients that will benefit from the use of immune checkpoint inhibitors. And now we move forward on the extractions of and the identifications, the calling of mutations directly from RNA-seq data, which is quite powerful to identify new epitopes, and especially when we combine that with existing algorithm that already describe gene fusion. So this will enable and disable and open the route for personalized therapies in these patients. And more recently, we decided also to address the questions of non-coding RNAs, because all what have been described so far was based on coding RNAs. So, and we have to understand that these non-coding RNAs are now more and more targeted with new genetic drugs. So I would like to thank you for your attention, and I would like to thank also my colleagues, so especially Jacques Collage, who is the team leader at IRCM of this bioinformatics group, and all the work on SDC and PCNSL tumors were performed by Melissa Alame during her PhD, and Simon Cabello performed the work on LRDP. And thank you again.
My name is Professor Mustafa Al-Awadi, a professor at the National Research Center in Cairo, Egypt. As a matter of fact, more than 200 vaccine candidates are now in the preclinical or clinical trials. 31 of these vaccines are getting into the clinical trials. 6 of them are at the final stages, phase 3. I would like also to add here that some of the technologies used to develop these vaccines have not been licensed for the use in humans before. This cartoon shows the structural organization of the virus, where the virus contains envelope, which is comprised of 3 proteins: a spike protein that is responsible for getting the virus into the host cell, and the membrane and the envelope. The genome of the virus is surrounded by a nucleocapsid, and the gene that codes for the spike protein is included within the entire genome of the virus. This picture shows the genomic structure of the whole virus, and here is the sequence of the S protein, the spike protein, that is comprised of 2 subunits, S1 and S2. Both subunits are connected by a junction sequence called the furin sensitive site, which is really unique among the rest of the coronavirus. Another important immunogenic part of the S1 subunit is the receptor binding domain, which contains a number of immunogenic sites, and it functions to allow the spike protein to get or to bind to the receptors on the host cell. Another important domain in the S1 protein is the NTD, N-terminal domain. How our immune system works for this virus. Coronavirus gets into the host cell via the binding between the spike protein and the angiotensin converting enzyme 2 receptor, ACE2 receptor. Once the virus particles get into the cytoplasm of the cell, it is circulated or ingested or engulfed by a lipid vesicle that allows the release of the viral genome that goes to the ribosomes and start translation of the viral proteins and the viral assembly. The viral particles get outside the cell by the exocytosis, and these viruses either go to infect other cells or they being trapped by the antigen presenting cells or the dendritic cells, where these cells express the viral proteins they have ingested to the cell surface, where they are recognized by the T helper cells of the host. These T helper cells, or CD4, they are converted to CD8 or the cytotoxic T lymphocytes. This is the T cell, the cellular immunity. These cytotoxic T lymphocytes, they destroy the cells that are infected with the viral particles. Another route for the T helper cells is the stimulation of the B cells to produce antibodies. Among them, there are some antibodies that can neutralize the virus and block its recognition. The technologies used to develop COVID-19 vaccines are four main different technologies: whole virus technology, viral vector technology, nucleic acid vaccines, or protein-based vaccines. Most of the trials going on right now in the whole world are protein-based, followed by the viral vector technology, followed by the nucleic acid, and by the end, the Chinese technology that is the whole virus vaccine. The first technology is the whole virus vaccine, either to use the weakened. This is the traditional vaccine, or the traditional technology to develop vaccines. This is the weakened, either weaken the virus or to inactivate the virus. Weakening the virus can be used by passaging the virus through cell culture in human or animal cells, so that the virus can acquire mutations that render the virus unable to produce proteins and become non-virulent or non-pathogenic. A company called Codagenix in Farmingdale, New York, in USA, is working using this technology. I would like also to add here that the new technology of CRISPR-Cas9, the genome editing technology, can be used to induce mutations that render the virus non-pathogenic. The other technology of the whole virus vaccines is the inactivated virus, either by using heat or by using formaldehyde. In either way, the whole virus, either weakened or inactivated, are used for vaccination, which will provoke the immune system to induce the production of the T and B cell response. So to give you some hints about this, 7 groups of the investigators all the world are working on this. A company called Sinvat Biotech in China is following this technology. Let me say that this is the technology used to develop the human vaccines known today, like polio, measles, mumps, rubella, chickenpox, and also the conventional influenza vaccines. One major disadvantage of this technology is it is non-economical to be used at a large scale in the industry to produce billions of doses, which we need now. The Chinese estimated the price per dose $140 per dose. The second technology is the viral vector vaccine, either by using the replicating viral vector, such as the weakened measles virus. They can use the measles viral genome as a vector, and they have already used this for Ebola vaccine, the newly approved, and also for HIV vaccine. Or use the non-replicating virus like adenovirus, and this is the main technology used by the British scientists in Oxford in association or in collaboration with AstraZeneca. The spike protein from the COVID-19 will be cloned in the whole genome of the vector virus, like measles or adenovirus, and it will be directly injected into the vaccine to provoke the immune system, or probably alternative way, to use these to transfect cells and allow the cells to express the spike protein of the coronavirus. Companies like CanSino, Johnson & Johnson, the giant company, and AstraZeneca are following these approaches. Johnson & Johnson already developed viral vector vaccines for rabies, HIV, Ebola. I would also to add that Sputnik 5, the Russian vaccine, is produced by this technology through the Russian Defense Ministry in association with Gamaleya company. And this is a similar technology to CanSino from China. The Russian people started already the phase 3 clinical trial using 40,000 people as volunteers. Also, the Oxford vaccine is following this technology from Oxford Vaccine Group in association with Jenner Institute from the same university, also in collaboration with AstraZeneca. They started their work on January 2020. They used the adenovirus vaccine or the adenovirus vector to produce the spike vaccine. They call it CH-ADOX1, CH from chimpanzee, adenovirus, Oxford 1. They have already started phase 3 in United Kingdom as well as outside the Great Britain, in India, South Africa, and Brazil. The third technology is the nucleic acid vaccine technology. Either using the RNA completely and directly. The RNA does not need any vectors because it is encased by lipid coats and it can get into the cells very easily, or convert the RNA to cDNA and consider it as a DNA vaccine. And in this case, it is different from the last technology, the viral vector vaccine, that it needs host cell or mammalian cell intermediate. In this case, most of the scientists use the HEK 293 human fetal cells, human embryonic kidney cells. It has been obtained from a legally aborted fetus, female fetus, like 60 years ago, and it has been in the business for about more than 5 decades, and it is very safe, and it allows the expression of the foreign proteins like the spike protein in this case. The purified protein can be purified from the cell debris and from the other components of the media, and it can be used as a subunit vaccine immediately for vaccination. The companies following this are mainly Inovio and Moderna virus from USA. Some vaccines used for veterinary use using this nucleic acid vaccine, like canine melanoma and the West Nile virus in horses. Let me tell you a few words about Moderna work. Their vaccine is called mRNA 1273 vaccine. The title of this vaccine, it is a lipid nanoparticle encapsulated, non-replicating nucleoside modified mRNA-based vaccine. This technology has been adopted also by other companies like Pfizer, BioNTech, and CureVac. This technology has been used for MERS vaccine before by some other companies. The estimated price for this per dose for this vaccine is about 32 to 73 US dollars. Moderna started the work in March 2020. They completed phase 1. They completed phase 2 by the end of July 2020, and they used in the phase 2 a non-human primate challenge study. So this was probably a unique experiment to challenge the monkeys with the viral infection, either vaccinated or not vaccinated. Also, they used a relatively small cohort from human volunteers, 300 young adults aging from 18 to 50 years, 50 old people from 56 to 70 years, and elderly people 70 years and above. They have already started phase 3 by the end of July 2020. Moderna is working in collaboration with the NIH from US and BARDA, which is the Biomedical Advanced Research and Development Authority. They plan to use 30,000 participants. They allowed a budget for almost $1 billion American dollars. The recommended dose is 100 microgram twice, 28 days apart. One of the major advantages of this RNA vaccine is that it is equally safe and effective for both adults and elderly people. The major disadvantage of this vaccine is its transportation and the storage, because it needs, RNA usually is a fragile molecule. It needs ultra low temperature. So they need liquid nitrogen or dry ice. So it might be a little problematic in transporting the vaccine from USA, where it is produced, to other parts of the world. The fourth technology is the protein-based vaccine, either subunit vaccine, and this is really very easy. It is, you can clone the spike protein from the coronavirus into a mammalian expression vector and allow this to be expressed and purified the subunit, the spike protein, in a pure form. And you can use it directly for vaccination. About 28 teams all over the world are following this. Only 5 teams are working on this technology, virus-like particle technology, in which they evacuated the coronavirus from its genome, so it is not, it becomes not pathogenic anymore, and you use the proteins around the envelope for provoking the immune system. However, this is difficult to manufacture, and it is a high-tech technology, but it is not really very practical technology. Now the landscape, most of the activities are private and industrial. Partners are working on this, followed by the academias, and followed by the about 5% only from the public and non-profit organization. Okay, this is our vaccine at the NRC that I have developed with my team. This is the S1, S2, the entire S protein. It is cloned in a bacterial vector. Of course, in this form, it cannot be used as a vaccine. We need to subclone this purified S gene from the coronavirus into another. We are still thinking about the best technology. We do not want to repeat other people's work. We can either clone it into viral vectors, or we can clone it into mammalian expression vector and use it for subunit vaccine production. We have not decided yet. I know that we are lag behind the others, but at least we have completed the most difficult part, which is cloning of the S gene. Thank you very much.
Welcome everybody. Today I'm going to give a talk about my my topic, which is the effect of hyperlipidemia on the donor-neuron disposition. First of all, I would like to thank Assyut University for their invitation. I'm so happy to be one of your invited and guest speaker. So I hope you will enjoy my talk and get some information about this, the drug and disease, like drug and disease interaction. First of all, what do we expect from this talk? So we have to understand the hyperlipidemia as disease, also to know the importance of studying the drug-disease interaction, and to consider different experimental studies related to the drug-disease interaction, and to understand the changes of neuron disposition in hyperlipidemia. So let's start with the hyperlipidemia as a definition. So there are many definitions for hyperlipidemia, but the American Heart Association, they define the hyperlipidemia as an elevation of one or more lipids, including lipoprotein, cholesterol, and triglyceride in the bloodstream. So this kind of disease, it might be caused by different factors. It might be genetic, diet, medical condition, and drugs. So when we talk about the hyperlipidemia, it's important to take a look to the important important parts of lipids, which is like lipoprotein. So lipoprotein, they are the packages by which the lipid movement is accomplished through the body. It's classified into chylomicrons, very low density lipoprotein, VLDL, low density lipoprotein, and high density lipoprotein. So in experimental and like at animal scale experiments, there are several experimental model of hyperlipidemia. Hyperlipidemia can be induced by giving high fat diet to the animal. The drawbacks of this kind of model, it takes longer time and it might cause inflammation to the animal species, which might not give a good data from this kind of model. And also the administrating a chemical compound like polyxemer like 407 or the tritone WR1339 or the genetically manipulated animal species. So on my talk, I'm using the experimental animal model of hyperlipidemia. I'm using the polyxemer 407. So this kind of model, it has like it causes like state of hyperlipidemia with a shorter time, around 24 hours. Also, the advantages of this model is reversible in nature, and it's like dose dependent and non-toxic. So when you increase the dose, so you expect higher level of lipids in the animal model. So as we like, I'm focusing on the drug of interest, which is the dronedarone. So let's talk a little bit about the cardiac arrhythmia. So cardiac arrhythmia simply it's a medical condition. It's characterized by the irregular rate of the heartbeat. So arrhythmia, the heartbeat too fast, which is kind of like it's called tachycardia, or it's slow like bradycardia. And there are like several medication and pharmacological agents used for the therapy of the arrhythmia. The widely used one, which is the amiodarone. So amiodarone is an important and often used antiarrhythmic agent. It's like first line therapy. It's by its nature, it's a highly lipophilic drug and it has several toxicity, which is the liver, lung, and thyroid toxicity. And this kind of toxicity, they are very serious and very dangerous to the patient. So amiodarone from the structure, as you see it, so it's like it has like a complex structure, and from the pharmacokinetics point of view, it has a major metabolite, which is this ethyl amiodarone, and also it's metabolized by several cytochrome B450 enzyme, which is the 3A4, 1A1, and 2C8. These are the main isoenzyme for the metabolism in the human. But for the rats, there are like several enzymes, for example, CYP3A1, 3A2, 1A1. These are the major enzymes involved in the rats. So my drug of interest, it's like a kind of, we can say a brother to the amiodarone. So dronedarone is a benzopurine derivative. It's similar to this, so it's kind of a brother to the amiodarone. It's a derivative of amiodarone. It has less lipophilicity. It's less toxic, and there is no iodine in its structure. So these three factors that might decrease the toxicity of this compound itself. So it's also used for the treatment of arrhythmia, but unfortunately it has there were several case reports about the pulmonary and hepatic toxicity in humans. So if we go back to the imidazole and make a comparison between the imidazole and dronedarone, so imidazole has like oral bioavailability around 33 to 65%. It has a reported plasma clearance around 2.63 ml/min per kilogram after giving IV dose. Human data suggests the volume distribution at steady state to be 33 to 65 liter per kilogram. It has a longer half-life, which is around varies like between 20 to 46 days, and also it has a high protein binding, which is more than 96%. So when we talk about dronedarone, it's like 70 to 90% of the dose absorbed. It has a low oral bioavailability around 44% in fasting, versus like 15% in feed state. Also, it has a reported plasma clearance around 38.5 ml/min per kilogram after giving an IV dose. So human data suggests that dronedarone volume distribution at steady state to be around 18 liter per kilogram. So this is what we know about dronedarone. Unfortunately, this is the only information we have. There is a little pharmacokinetics data and peer-reviewed literature. So most of the data from the product monograph. So as we mentioned before, so it's important, like what we got from the monograph, the dronedarone metabolized by cytochrome B450. It's similar to the imidazole. It's like by CYP3A4 or 3A5. This butyl dronedarone is the metabolite of dronedarone with less anti-arrhythmic effect, and it has shorter half-life compared to the imidazole, which is around, in case of dronedarone, it's like 24 hours. So what we know about the effect of hyperlipidemia and on the pharmacokinetics of imidazole. So I was lucky to work in a lab which was focused on the imidazole. So in our lab, they found that the hyperlipidemia in animal species model caused some changes to the imidazole, which include decreasing the volume distribution and clearance, decreasing the plasma unbound fraction, and also alter the tissue distributions of the imidazole in different organs. Also decrease the hepatic cytochrome B450. So our rationale for this study for the effect of dronedarone. So dronedarone is a lipophilic drug. It's an imidazole derivative. So there were several cases reported. It has a pulmonary toxicity and also hepatic toxicity. So we expect that we have the same behavior like dronedarone in hyperlipidemic state. So we hypothesize that having like pharmacokinetics parameters in rats similar to that and similar to those of imidazole, so exhibit differences in pharmacokinetics in normal lipidemic and hyperlipidemic rats. So, but we got some problems. Unfortunately, there were little pharmacokinetic studies in the human. We only have the manufacturing data, no peer review, no pharmacokinetics data in rats, and there is also analytically we don't have enough or a clear analytical method to measure the levels of dronedarone in biological sample. So the first objective of our project was to develop a sensitive and validated reverse phase HPLC for measuring the dronedarone, and also studying the effect of the pharmacokinetics of dronedarone in normal lipidemic, which is the normal state, and also in hyperlipidemic state, also to assess the tissue distributions of the volume to assess the effect of hyperlipidemia on the tissue distributions of dronedarone in rats. So we started with the first objective. We got a good data about the analytical method. We got an excellent linear relationship. So we got an excellent analytical method with a recovery more than 80%, with also a very sensitive method. It measured the concentration started from 25 to 1,000 nanogram per ml. Also, the lower limit quantification around 25 nanogram per ml based on 100 microliter of rat plasma. So this is the chromatogram, which shows our drug of interest. So we have different chromatograms here. As you see, we have the rat spike with 250 nanogram, and also we have a blank plasma, and we have samples collected from the rat. So we published this data in Biomedical Chromatography journal. So now we will move on to another study, which is studying the pharmacokinetics of the dronedarone. So we have the rats. So we give them a dose as IV, oral, and intraperitoneal. We collected the samples at different time points, and we measured the concentration by a newly developed HPLC method. So we found out this is the mean plasma concentration versus time of different routes of administration. So as we give it IV, intraperitoneal, and oral. So from this figure, we can conclude that, and we measure the different pharmacokinetics parameter. So we found out the clearance of dronedarone. It's similar to the amiodarone. For the plasma distribution, we found out we have for the volume distribution at steady state, we have the level of and the value of volume distribution at steady state for dronedarone is less than as in the amiodarone in animal model. For the half-life, we found out it has a shorter half-life compared to the amiodarone. For the bioavailability, you found out also dronedarone has a lower or low bioavailability compared to the amiodarone. For the data we got from our animal study, if we compare that to the manufacturer monograph, we found out in rat we have a slower clearance, which is around 25 ml per minute per kilogram, versus which was reported in the product monograph, which is around 38.5. Also, we have in rat for the volume distribution at steady state, we found out it has a smaller VSS value compared to the human. Regarding the half-life, we can conclude that we have a shorter half-life in rat compared to the human data, which was reported in the product monograph. So and we move from based on this one, both on the previous study. So as we have a clear data about the pharmacokinetics parameters values in the healthy and control and normal rats, so we move on to the affecting the hype. So it's good to know what changes happened when we use our model, which is of hyperlipidemia. So we have a rats, and we use our model, which is the poloxamine. We give the rats a dose of 1 gram per kilogram. This is for the treatment group, and for the normal group, we give them saline IT. So we waited for 36 hours. Then we make sure we have a hyperlipidemic rat. We can examine that by collecting, I feel some volume of the blood, and we can see if there is changes in the changes in the plasma lipids. We can notice that by the color. If we collect the sample, then we centrifugate it, then we can see if there is a lipid or not by having a white clear white or dark, like a white plasma. So we can conclude this rat is hyperlipidemic. So after that, we give a dose of dronedarone as oral. Then we collect samples at different time points. So we got this a beautiful figure. So as you see, we have a higher concentrations of dronedarone in hyperlipidemic rats compared to the plasma. So we have, this is what we got from the data we have. So we can, based on that figure, we calculate different pharmacokinetics parameter. We found out this in hyperlipidemic rats, they have higher exposure of the drug, which can be explained by high and larger value of larger value of area under the care compared to the normal group. For also, this is for the from time 0 to 24 hours, and this is for the infinity. And also, we found out the half-life also, like it's the same for both the normal lipidemic and hyperlipidemic. So we published all the data we have in the Biopharmaceutics and Drug Disposition journal. It was like a very interesting data about the effect of hyperlipidemia on the dronedarone pharmacokinetics. So we move on to another study, which is the distribution of dronedarone in different tissues. So we conducted the same study, but using different animals for sure. So as I mentioned before, we give them a Voloximab dose or the saline dose, then we waited 36 hours, then we collected the samples at different time points after giving an oral dose. So we have like around 4 rats per time point, and also we collected the heart, liver, lung, and we analyzed the concentration of dronedarone in this kind of tissue. So we found out that dronedarone was detected and measured in all species at quantifiable concentration in all tissue. As you see from this figure, so we have highest concentration of dronedarone in the control group. It was the highest in the heart, then followed by the lung, followed by the liver, then in the plasma. For the hyperlipidemic rats, we found out that the highest concentration was in the lung, the highest concentration in the lung, followed by the heart, then followed by the liver, then followed by the plasma. So based on the data we have and on the experiments we have, so we had a rapid, simple and validated HPLC method for quantification of dronedarone. Also, this method can measure as low as 25 nanogram per ml using a small volume of plasma. This kind of method can be used in dronedarone pharmacokinetics studies. Also, we found out in normal lipidemic rats, we have a poor bioavailability, which is less than 20%. And also, we found out dronedarone is extensively distributed with high volume of distribution among different tissue. Also, we found out the hyperlipidemia has an effect on the pharmacokinetics of dronedarone, and it might be cause direct effect on the metabolizing enzymes or the transport protein. So in heart, which is the site of action of the drug, dronedarone has the highest concentration, which is expected. In contrast, lung has the highest concentration of dronedarone in hyperlipidemic rats. So we can, assuming that depends on the transporters involved and distributed at different different organs, we might expect different concentration and different response at different kind of tissues. So based on these studies, we can say it's very important to consider pharmacokinetic changes in disease states as in hyperlipidemic model, experimental model, also based on the data we have. So we might need to do some changes to the dosage if we are giving like in clinical setting, and also very important to explore the human data regarding the dronedarone in healthy patient and compare that to the hyperlipidemic patient, which is like very wide in our modern society due to the changes in the diet and lifestyle. So we expect some changes in pharmacokinetics parameter, especially area under the curve or the volume distribution or the clearance or half-life among different subjects, like healthy or hyperlipidemic patient or any patient who are in different medication. It might be change the pharmacokinetic parameter change if he has a different diseases. So this is the important points about my talk. At the end, I would like to thank King Saud University Scholarship for giving me a chance to do this kind of experiment. Also, I would like to my mentors, Professor Diane Brooks and Professor Simon Kavi, who were with me in my studies, and also special thanks to the University of Alberta for giving me a chance to be one of graduate students in their campus in Edmonton. Thank you so much for your time. I hope you enjoy my talk. I'm open for any questions. Thank you so much.
بسم الله الرحمن الرحيم. السلام عليكم زملاء الأفاضل. إن شاء الله محاضرتنا النهاردة هتكون على Nano Radiopharmacy Approach for Tumor Theranosis. ده approach جديد لتيومر ثيرانوزس، وخلال المحاضرة هنفهم بالظبط تيومر ثيرانوزس مقصود بيها إيه، وهو بيزد على ميكس ما بين النانو تكنولوجي والنيوكليير تكنولوجي. في البداية أنا اسمي تامر مصطفى صقر، Associate Professor of Radiopharmaceutical Chemistry and QC Manager in Radio Isotope Production Factory في هيئة الطاقة الذرية المصرية. منوط بينا إنتاج المستحضرات الصيدلانية المشعة لتغطية القطر المصري بشكل كامل. التوبيك الأساسي الخاص بالمحاضرة دي أنا فيه في كوبريشن مع الورلد نيوكليير يونيفرسيتي في إنجلترا، ميزوري يونيفرسيتي في USA، والانترناشونال اتوميك انرجي ايجنسي مع اليونايتد نيشن UN. الفكرة ببساطة إن إحنا بنخلط نوعين من التكنولوجي مع بعض، النانو تكنولوجي والنيوكليير تكنولوجي، to try to achieve approach جديد لعلاج وتشخيص التيومر بشكل عام. عشان هنبتدي نفهم بعض البيزكس الخاصة بالنيوكليير فارماسي وبعض البيزكس للنانو ميديسن، ونبتدي نفهم ازاي عملنا الميرج ده. نيوكليير فارماسي هي ببساطة ان انا بعمل drug in prescription based على radioactive atom واستخدم الكيمستري ما بين carrier وراديو ايزوتوب to prepare radiopharmaceutical agent to be injected inside the body علشان اقدر اوصل للدياجنوزس او treatment للديفرنت body organs. في البدايه هنشوف مع بعض فكره النيوكلير فارماسي من خلال الفيديو ده. يبقى من خلال الفيديو ده قدرنا نشوف ان انا بقى عندي كيميكال ستراكشر. في الكيميكال ستراكشر ده ببتدي ان انا اعمل insertion ل radioactive atom to be part of this molecule، وبعد كده احوله ل dosage form to be administered، وبعدين يبتدي الراديواكتيف مولكيول يجو through all over the body علشان يتراكم في target organ اللي احنا عملنا له designing من البداية، ويتراكم الراديواكتيف pharmaceutical agent inside the target organ to emit alpha أو بيتا أو جاما. وبيزد على الألفا أو بيتا أو جاما ببتدي أستخدم الراديو pharmaceutical agent ده يا إما للثيرابي يا إما للدياجنوز. وبكده هنقول إن إحنا عندنا mainly 3 types of radiation: alpha, beta and gamma. الفرق الأساسي ما بين الألفا والبيتا مقارنة بالجاما إن الألفا والبيتا are particles. So we have some sort of collision between these particles and the DNA inside the nucleus of the atom. وبالتالي الألفا والبيتا particles are used mainly for the therapy. لكن gamma radiation is an electromagnetic wave عندها مقدرة عالية جدا إن هي تعمل penetration outside the body، وبالتالي mainly بتستخدم في الإيميجينج. فبنقول إن النيوكلير ميديسن هو عبارة عن الموداليتي أو البرانش من الميديسن اللي بيستخدم الراديو ايزوتوب أو راديو نيوكلايد والفينومينا خاصة بالراديو اكتيف ديكاي to diagnose أو treat different body organs أو diseases. وبالتالي النيوكلير ميديسن بيستخدم الراديو pharmaceutical agent to diagnose أو treat different diseases. فبنشوف الفرق الأساسي ما بين الراديوجرافي والنيوكلير ميديسن. في الصورة اللي على الشمال في الإكس راي أو الراديوجرافي الراديو اكتيف سورس أو السورس للإشعاع بيكون from outside the body. وبعدين الريز اللي هي الإكس راي بتدي penetrate the body وتتجمع على فيلم. الفيلم ده يديني opaque picture. الأوبيك بيكتشر ديت ابتدي اقرا منها المورفولوجيكال شيب السايز of different body organs. لكن في حالة الجاما راي اللي هو النيوكلير ميديسن لا، زي ما شفنا في الفيديو بيتم حقن الراديو اكتيف ماتيريال داخل البودي، وبالتالي بعد السيركيليشن بيتحول البودي نفسه as a radioactive source، يعمل emission للجاما راي، وبالتالي ابتدي استقبلها على الجاما كاميرا ديتكتور. وبكده بقول ان انا بمجرد ان انا بحقن الراديو اكتيف ماتيريال انسايد ذا بودي بتبتدي تروح التارجت اورجان زي ما شفنا في الفيديو. لو اعتبرنا ان الراديو فارماسيوتيكال ايجنت هنا هيتراكم في الهارت، اصبح الهارت نفسه as a radioactive source يemit radiation اللي بيتجمع بالجاما كاميرا علشان يديني morphological picture للأورجان beside function. فلو أنا عندي biological target في أورجان معين سواء انزيم أو ريسبتور، ببتدي أختار biological active molecule. البيولوجيكال active molecule هو drug سواء agonist أو antagonist capable إن هو يعمل attachment بشكل selective على الريسبتور اللي موجود في التارجت أورجان، وابتدي باستخدام الراديو سينثيسيس ميثودز اربط الراديو اكتيف اتوم اللي هي الراديو ايزوتوب مع البيولوجيكال اكتيف موليكيول، وبكده يبقى الجزء الاخضر ده كله هو الراديو فارماسيوتيكال ايجنت اللي بيتم حقنه داخل البادي. فلو بصينا في الصوره اللي على الشمال هنلاقي ان الراديو فارماسيوتيكال ايجنت ابتدى يمشي داخل البادي، يتراكم في الديفرنت بادي اورجانز ب some sort of selectivity according to design اللي انا عامل بيه الراديو فارماسيوتيكال ايجنت. لكن اكبر ميزه هنا لو انا حقنت ماده التارجت اورجان بالنسبه لها الكانسر، هنلاقي ان الراديو فارماسيوتيكال ايجنت ابتدى يتراكم في كل الاماكن اللي فيها ولو عدد بسيط جدا من السيلز اللي initiate tumor نتيجة الميتاستاسيس، وبالتالي اداني مسح كامل للبودي بمجرد ان انا عملت راديو فارماسيوتيكال ايجنت فيري سيلكتيف للتيومر. بعد عمليات الحقن ببتدي اصور البيشنت على الجاما كاميرا ويديني 3D بيكتشر زي اللي احنا شايفينه. وبكده بنلاقي ان النيوكليير فارماسي بيديني 5 ميزات اساسيه. اول حاجه نون انفيزيف تكنيك. اذا كان الطبيب محتاج ان يعمل سيرجري عشان يبتدي يشوف داخل البادي ايه المشكله، لا انا بديله ايجنت قادر على اني يخلي الطبيب يشوف ايه اللي داخل البادي من غير ما يحتاج لسيرجري، فبنقول عليها نون انفيزيف تكنيك. بيسايد ان بيديني المورفولوجيكال شيب، كل اورجانز سايز قد ايه داخل البادي، بجانب زي ما شفنا في صوره الهارت مديني ديفرنشيشن ما بين الديفرنت سيلز اللي موجوده في الهارت، وبالتالي بيديني الاورجان فانكشن. في الصوره البيكتشر اللي بتدور بيبتدي يعمل لي هول بادي سكان، بيمسح البادي كله، يبتدي يخليني ابتدي اشوف واقيم الكلينيكال كيس. فلو عندي تيومر في اورجان، لو الفيزيشن هيبتدي ياخد قرار ان يشيل التيومر، لو عملت راديو اكتيف سكان مسح ذري هبتدي ان انا اشوف هل في اي نيوكليوس موجوده to start tumor in different organs، وبالتالي القرار ممكن يختلف خالص لان انا هنا قيمت هل في ميتاستاسيس ولا لا. خامس ادفانس موجوده هي ان انا اقدر استخدم one agent for therapy and imaging اللي احنا بنقول عليها ثيرانوزس اللي كانت موجوده في تايتل تيومر ثيرانوزس. يبقى التيومر ثيرانوزس معناها ان انا بعمل one agent يديني جاما، ابتدي ان انا اعمل ايميجنج ودياجنوزس للكيس، ويطلع او يعمل ايميشن لالفا او بيتا، وبالتالي يستخدم في حاله الثيرابي. فبكده فهمنا الابروتش الاولاني خالص اللي هو فكره النيوكلير ميديسن او النيوكلير فارماسي او النيوكلير تكنولوجي بشكل عام. الكونسيبت الثاني هو النانو ميديسن. النانو ميديسن يتقال عليه ان هو دلوقتي الفيوتشر اوف ميديسن. ليه؟ لو ابتدينا برضو نشوف بداية الفيديو ده،
such as antibodies, the hydrophilic bonds.
لو انا
This
حضرت
polymer
نانو
coating
بارتيكل زي اللي
can be
قدامنا،
subsequently surface modified with various cancer targeting ligands such as antibodies or small molecules. The nanoparticle solution can then be modified into a form to allow for intravenous administration. The nanoparticles are quickly distributed throughout the body by the circulatory system, with which they are also delivered to the site of the tumor. Times smaller than red blood cells, nanoparticles possess the unique ability to permeate through the leaky walls of tumor vasculature, which are formed during the process of tumor growth.
هنا في الفيديو انا شفت ان مع تحضير النانو بارتيكل وفي وجود التيومر بيديني ليكي فاسكلاتشر، وبالتالي زي الاستيك لو انا شديت الاستيك بيبتدي البور، لو في موجود بور معينه في السايز يزيد. فبنلاقي ان في حاله التيومر البور سايز بيكون 150 نانومتر، لكن في النورمال سيل 5 نانومتر. فهنقول ان النانو ميديسن هو استخدام سايز في around 10 to the power -9. فبنقول ان الدي ان اي diameter 2 نانومتر، السمولست بكتيريا 200 نانومتر. طب ايه ميزه النانو ميديسن؟ النانو ميديسن هو بيقدر يخليني اتارجت السيلز او التيشو على السيلولار ليفل. يبقى انا بحاول ان انا اعمل دراج to treat in the same level مع الديسيز. فالنانو ميديسن بخلط ما بين 3 ساينسز، نانو تكنولوجي مع الكيمستري مع البيولوجي، وبالتالي اطلع برودكت يقدر يعمل مونيتورنج، ريبيرنج، كونستراكتنج، كنترولنج السيلولار والموليكولار ليفل. بما ان الديسيز هو اصلا دامج على السيلولار والموليكولار ليفل، يبقى انا كده كاونتر اكت الديسيز ان ذا سيم ليفل، وبالتالي هيديني كفاءة الدياجنوزس، كفاءة الثيرابي، ويقلل لي بشكل كبير السايد افكت. لو جينا بصينا داخل البودي زي ما شفنا في الفيديو هنلاقي ان النانو بارتيكل smaller than الار بي سيز، وبالتالي الموفمنت داخل البودي برغم ان هو بارتيكل it's moved freely in blood circulation. فيقدر يديني small size، ياخد كميه كبيره جدا من الكونسنتريشن للدراج، واقدر اعمل له ديزايننج ان يبقى تارجت سبيسيفيك. يبقى انا عندي بارتيكل واحده فيها كميه كبيره جدا من المولكيولز بنقول عليها البيلود هاي كونسنتريشن من الدراج، بجانب ان انا اخليها تارجت سبيسيفيك. طب ده هيفيدني بايه؟ هيديني مولكيولار تارجتنج عالي، دراج ديليفري عالي، less side effect، و improved detection. طب لو خدنا الميزه دي وابتدينا نطبقها على الكانسر، لما نيجي نشوف الكانسر هنلاقي زي ما قلنا في النورمال سيل البور سايز ما بين السيلز 5 نانومتر، انما في المكان اللي فيه كانسر البور سايز ما بين السيلز 150 نانومتر، وبالتالي الليكي فاسكولاتشر اللي موجود في البلاد فيزلز داخل التيومر هتبقى بسهوله جدا النانو بارتيكل هيقدر ان هو يعمل باسيف تارجتنج للتيومر. فبنقول عندنا فينومينا اسمها enhanced permeability and retention. مقصود بيها ان انا طالما هلاقي النانو بارتيكل داخل البودي لما يبقى beside the normal tissue it will not penetrate out of the blood، لكن مجرد ان هو يوصل للليكي فاسكولاتشر اللي موجود فيه الكانسر هيحصل له go out from the blood من الليكي فاسكولاتشر دوت ويتراكم داخل الايه؟ الكانسر تيشو، وبالتالي هيبتدي البارتيكل نفسه يحصل له engulfment داخل السيل، وبالتالي يديني high concentration of the drug inside the cancer directly. وبالتالي لو احنا خلطنا فكرة النانو مع النيوكلير هيديني ايه؟ هيديني بارتكل فيها راديو اكتيف ماتيريال. الراديو اكتيف ماتيريال قادره انها تعمل لي ايميجنج وثيرابي وبالتالي ثيرانوزس. ويقدر النانو بارتكل ده يشيل كميه كبيره جدا من الراديو اكتيف ماتيريال اللي بنقول عليها البيلود، ويقدر يقلل لي الليكج بشكل عام وبالتالي يقلل السايد افكت. فلو بصينا للكانسر بشكل عام هنلاقي ان في كانسر ستيم سيل، كانسر سيلز، نورمال سيلز، مالجنانت سيلز، ميوتيتد سيلز. لو انا استخدمت الراديو اكتيف ماتيريال داخل التيومر بشكل مباشر وتراكمت داخل التيومر نتيجه الراديو اكتيفيتي اللي موجوده، هيعلي لي بشكل كبير جدا البنتريشن بانه يوصل لكل السيلز اللي موجوده داخل التيومر. وبالتالي لو انا عندي تيومر وبعالجه بكيموثيرابي، دايما الكيموثيرابي هيجي للبريفرال بارت. طول ما هو بيجي في البريفرال بارت هيفضل دايما الكانسر ستيم سيل الموجوده داخل النيوكليوس جوه الكانسر موجوده، وبالتالي الجروث هيحصل تاني. لكن في حاله البنتريشن العالي اللي ناتج من الراديو اكتيفيتي او الاشعاع اللي طالع من البارتكل، هقدر بسهوله جدا ان انا اوصل للجزء الخاص بالكانسر ستيم سيل، وبالتالي هقدر اوصل للتيومر هيلينج. ف جت الامم المتحده اليو ان ديزايند جلوبال بروجكت. جلوبال بروجكت ده عباره عن ريسيرش كوردينيتد ريسيرش بروجكت. الكوردينيتد ريسيرش بروجكت ده كان بين 13 دوله على مستوى العالم، بان احنا نديزاين النيو ساينس دوت بحيث ان احنا نطلع بالابروتش اللي يديني افيكاسي عاليه جدا، تارجتنج ابيليتي عاليه جدا للكانسر، وفي نفس الوقت يقلل لي السايد افكت بشكل كبير جدا. تعاون فيه 13 انستيتيوت على مستوى العالم، زي ما قلنا من 13 دوله على مستوى العالم، وكانت النتائج جيده جدا جدا. هناخد اكزامبل سريع للراديو اكتيف جولد نانو بارتكل. الراديو اكتيف جولد نانو بارتكل مجرد ان هي بتتحقن بتتراكم بشكل كبير جدا داخل الكانسر سيلز، نتيجه زي ما قلنا الانهانسد بيرميابيليتي اند ريتنشن. فبالفينومينا الاكيوميليشن العالي دوت ونتيجه الاميشن اللي طالع من الجولد نانو بارتكل اللي هو بيميت الفا وبيميت بيتا وبيميت جاما. البيتا مسؤول عن الثيرابي، الجاما مسؤول عن الايميجنج والدياجنوستس. فاحنا حضرنا باي جرين نانو تكنولوجي، استخدمنا فايتوكيميكال اي جي سي جي عشان نكون الاي جي سي جي جولد نانو بارتكل، راديو اكتيف جولد نانو بارتكل. وعملنا كاركترايزيشن وابتدينا نستخدم دوت في الكانسر تريتمنت وكمان في التريتمنت الخاص بالانفلاميشن. وبالتالي وصلنا في الاخر لكفاءه عاليه جدا تقابل 85% هيلينج في البروستيت كانسر في الدوجز. واحنا دلوقتي في مرحله نقل الجزئيه ديت للكلينيكال ترايل. الكلينيكال ترايل we are preparing a program هيتم في دوله البرازيل، هو كوبريشن ما بين مصر وما بين يو اس وما بين البرازيل، في ان احنا نحول البرودكت اللي احنا اتكلمنا عليه دوت للتقييم فيز زيرو في الهيومن ان شاء الله. بجانب ان كان في بعض البروجكتس اللي هي going on مش بيزد على الجولد بس، بيزد على الاير، بيزد على سيلفر، بيزد على different radio isotopes. وابتدينا نحط newest approach ده لسه السنه اللي فاتت ان احنا نعمل النانو بارتكل ديكوريتد بالتارجت مويتي، بحيث ان احنا نستخدم الباسيف تارجتنج بلس الاكتيف تارجتنج. وبالتالي الكونكلوجن العام او التيك اواي key points ان دايما merging between different sciences can solve many of the challenging points. زي ما احنا خلطنا النانو تكنولوجي او النانو ميديسن مع النيوكلير تكنولوجي، وبالتالي كان open new approaches في الساينس. some challenging questions. ايه رايك كساينتست؟ هل فعلا فكره merge between the two different technologies دول النانو مع الراديو هيديني فعلا كفاءة للكانسر تريتمنت؟ وايه هي الصعوبات اللي احنا ممكن نواجهها؟ دي الريفرنسز للشغل المعمول على مستوى العالم، ودي اكسترا ريدنج. دول 3 كتب فنية من اليو ان في التخصص ده. ال 13 دولة احنا متشاركين كلنا في كتابة ال3 كتب دول. وشكرا جزيلا. السلام عليكم ورحمة الله وبركاته.
السلام عليكم. الحقيقة احنا قضينا وقت كويس جدا ومثمر مع الليكتشرز بتاعت السيشن ده، كانت سيشنز كويسة جدا. لو معانا بروفيسور إيمانويل كورنيلو معانا.
Yes, Cornelio.
you have been with us. We are enjoyed your lecture. I have a small question, Professor Cornello. Can we use this transcriptomic data in avoiding some rare congenital diseases in fetus?
Indeed, yes, transcriptomic data can be applied to any tissue. It's also applied in cell culture to identify drugs. We have many databases providing signatures of differential expressed genes in different conditions, influencing globally the expression of other genes in the genome. So this is a general approach. What I presented today is our pipeline developed for the study of tumors.
Okay, thank you. دكتور مصطفى معانا؟ لا.
كنا عايزين نوجه له سؤال بس غالبا مش موجود.
دكتور يوسف
دكتور يوسف موجود معانا. دكتور يوسف
اهلا بيك. عملت لنا محاضرة محترمة جدا. دكتور يوسف اتفضل يا دكتور هشام.
شكرا دكتور يوسف على المحاضرة الرائعة اللي حضرتك ألقيتها علينا اليوم. هو بس كان في سؤال لحضرتك وضحت لنا في السلايدز ان في حالة الهايبر لايبيديميا بيحصل accumulation للدراج اللي هو الدروندرون في اللنج، بيبقى موجود مينلي اكتر في اللنج كومبيرد بالاذر اورجانز او كومبيرد بالهارت. يعني هل في هنا هو تاثير ليه على اللنج؟ لانه في الحاله دي هيبقى موجود في اللنج مش موجود في الهارت اللي هي السايت اوف اكشن بتاعه زي ما حضرتك ذكرت. والجزء الثاني برضه في يعني في حاله الهايبر لايبيديميا او في حاله حضرتك قلت ان احنا لازم نعمل ادجستمنت للدوز فاينلي في حاله الهايبر لايبيديميا او لما يكون في اي حاله مرضيه بتصيب الانسان. هل حضرتك عملت ادجستمنت هنا للدوز من خلال الاستادي اللي انتوا عملتوها ولا ايه بالظبط؟ شكرا يا دكتور. اسمعني ولا
حلو قوي. انا لازم
الصوت
is off.
هاي دكتور تسمعني؟
سامعين حضرتك.
نعم.
اتفضل.
ممتاز. اول شيء شكرا جزيلا على صراحه المؤتمر هذا. كان ودي صراحه اكون في اسيوط والتقي فيكم شخصا بشخص، لكن الظروف سبحان الله ان شاء الله بستعد لازم على خير. بالنسبه لسؤالك يا دكتور، السؤال الاول اللي هو اللي كان عن موضوع الكونسنتريشن العالي في طبعا الاستدي اللي عملناه احنا كان الدوز it was equal for both healthy and the hyperlipidemic rats. But يعني we can find from the study زيادة الكونسنتريشن في اللنج it might be explain some side effects which it might be happened in the rats. So for your question, the first one, yes, we found some concentrations in the lung and the heart. In the heart is expected because it's the site of action, but for the lung we need more study to explore that action. So in this case, if we are expecting any side effects or like lung toxicity in human, we can be explained as a side effect of this drug. The second question for the dose was equal for both healthy and the hyperlipidemic rats.
Okay.
I have another question for you, Dr. Yusuf. معلش هنتعبك معانا معلش.
لا ايش دعوه، انا موجود لك ولكم دكتور.
We know that it is reported that fat meal increase the bioavailability of your drug, Dondarone. Would you suggest a mechanism? And also, I would like to ask. مش معانا ولا ايه؟
موجود كمان.
I would like to ask about the large number of metabolites which are inactive from the drug and the high volume of distribution. Is these 2 points are not disadvantageous for this drug, this medication?
We can you asked me 2 questions for the volume distribution. So we expect it's kind of disadvantage because it might the rats will be exposed to more side effects in this case because the drug itself, it will go deep into the tissue. So it we might expect some like side effects due to the volume of distribution. And for the bioavailability, it was reported that in humans, if the human take the medication or the dronedarone itself with the high fat meal, so the absorption will be increased and the bioavailability will be increased. But we haven't examined this in the rats. So our model, it's kind of like it's a model of hyperlipidemia. But for our study, we examined the pharmacokinetics profile and the fasted rats. So we haven't studied any effect of the food in that.
Okay, شكرا.
شكرا دكتور يوسف.
الدكتور تامر. دكتور تامر اهلا بيك. نورت. شكرا على المحاضرة القيمة. طبعا الثيرانوزس من الحاجات المهمة جدا ان حضرتك بيبقى فيه الدياجنوزس والثيرابي، فالدوز بتبقى تailored على البيشنت مظبوط جدا. بس انا ليا سؤال في النانو سايز دراج sometimes needs many steps for reaching the final preparation. I think this needs special radiation protection measures in this case. Because I am interested in the protection of the workers and the patient from excessive radiation. But when I make radio pharmaceutical nano size, I think the measures will be more restricted. You have the same opinion or not?
Firstly, I'm not totally with this opinion. Because indeed, iodine 131 is the drug of choice for the thyroid cancer. Yes?
Yeah.
Also, it's beta emitter and gamma emitter in the same preparation, and it is for therapy and imaging for the thyroid. Yes?
Yeah.
So the idea here of this theranostic agent that we use the ability of the nanomedicine to reach passively and retain effectively in the cancer cells to carry less amount of the radioactivity. So here we are using this nanomedicine approach to decrease the radiation dose which will be administered by the patient. This is the first point. The second point, the type of the energy of the emission. Because if we are talking about beta emission or gamma emission, each emission has its own energy. Yeah. To be effective for gamma emission, we are talking about from 150 kilo electron volt to about 300 or 400 kilo electron volt. At this range, you take the benefit issue from the radiation and avoid the toxicity for the patient. This is why we already choose gold 198 as a radioisotope of choice to prepare the nanoparticle based off it. This is the first point. The second point here, we avoid intensively the toxicity related to the gold nanoparticle itself, because we use here in the radio, we are always use massless amount of the drug, because we are dealing just with number of atoms. The pharmacological tool here is radiation, not the mass of the drug.
Yeah.
So if we merge it, both of the advantage of the radio with the advantage of the nano together in just one preparation, we can achieve some levels beyond what we are still here now. The idea also of our preparation that we already finished for the treatment of the prostate cancer in dogs, we are trying to inject the prepared nanoparticle intratumor directly. Intratumor injection in the prostate of the dogs. The idea here that you trying to decrease the injected dose, beside you gain a lot of retention time inside the cancer. You got the answer here? It's okay?
It's okay, but I still have two small points.
Yes, okay.
You say that the advantage of using the nano-sized radiopharmaceutical is to increase the dose or the concentration of the drug. But my aim in dealing with radiopharmaceutical is to give the minimum amount, the minimum to be effective or to explore, to diagnose something. So we don't need a high dose of a radiopharmaceutical. This is number one. Second point, have you performed any in vivo experiments?
Okay, for the first one, we said that we have a payload phenomenon. Yes? Payload means that if I have injected 1,000 molecules of methotrexate inside the body, we can say that just 10 molecules will reach the cancer cell. Yes?
Yeah.
So 990 will be toxic material inside the body. But here, I make the particle carrying about 20 molecules on it to direct it between the conventional radiopharmaceutical and the nano radiopharmaceutical. But the idea here is that we use of nano to inject the patient less amount of radiation.
Yeah.
This is what we need here.
Okay.
But if you are going to inject iodine-131 as a conventional or as nano, you will get the same toxicity if there are any toxicity.
Okay.
Okay.
Thank you very much.
شكرا.
Thanks a lot.
شكرا.
Thank you.
شكرا.
في النهاية
في نهاية السيشن بنشكركم جدا على حضوركم معنا واستماعكم. الحقيقة المحاضرات قيمة جدا، وبنتمنى انكم تواصلوا معنا لغاية ما ننهي مؤتمرنا بإذن الله غدا إن شاء الله. السلام عليكم.
شكرا جزيلا. السلام عليكم.